So good evening everyone. In our last class we have been discussing about electrophoresis, and initially we have discussed about this capillary electrophoresis technique, and we have seen how it is done, and we have also discussed that within a liquid, if we are having a mixture of ions, and if we place it under the electric field, how they will be distributed based upon the size and the charge. Okay, size is again, it is associated with the mass. Okay, then we have also discussed about the electrophoretic mobility mu, which is basically associated with the charge is to size ratio, and depending upon that, this movement of the ions, it happens within the electrocapillary system. Then we have also discussed about the mechanics of how this electrophoresis happens within a fused silica gel capillary system. Okay, and we have seen that there is a formation of electrical double layer, and then within that electrical double layer there is a fixed layer of the cation, and then again there is a mobile layer of the cation. And what is the advantage of using this capillary electrophoresis system? We have seen that under normal laminar flow, you will be having a parabolic pathway, and that leads to the broadening of the peak while you are analyzing the analytes at the exit of the capillary tube. How in the electroosmotic flow, when you apply this capillary electrophoresis, we get a flat front, and this allows you to get the analytes in a very short time, and you get a very sharp peak. So the resolution of the separation of the analyte, it is much better when we apply the capillary electrophoresis method. Okay. Then we have discussed about this system, then capillary electrophoresis method, how it is done, and all these things. We were not having time, so we could not discuss this one in the last class. So what happens is that we have discussed that the detectors, they are basically made up of they are basically employing the UV visible spectrometry and other types of spectrometric technique. Now in all of this, we have seen that they are basically dependent on the absorption, and we know that absorption, it is given by epsilon Bc. Okay, where C is the concentration, B is the this path length or the length of the cuvette, and eta epsilon, it is basically the molar extinction coefficient, and it is associated with the properties of your molecule or the analyte which you are analyzing. So over here we cannot change the concentration, because in a complex mixture, in how much quantity the concentration of the analyte will be present, we do not have any idea. Okay, molar extinction coefficient we cannot change. However, what we can change the path length. So this is the first one. It is one of the way to increase the path length. So what we do, this type of cell, it is regarded as the bubble cell. So what it is doing, from one end, your what you call eluate, it is coming in and it is entering into this cell, and over here we have physically increased the path length. If you see, the light is coming from here, so the distance from here to here, it is b. So we have artificially increased the path length over here, and because of this reason, we will be getting a greater absorbance. Okay. Secondly, so this type of cell, it is regarded as the bubble cell. Okay, and the second type of cell, it is called the Z cell. Over here we are not increasing any bubble. What we are doing, we are just changing the path. So now from here, what would be your path length? The path length would be somewhere in like this. So the distance between this to this point, say A to B, or let us say capital A to capital B, is equal to small b. So in this way we can increase the path length, and accordingly we will be having an increased absorbance. Okay. So if you see, we have initially discussed we can use UV spectrometry, we can use IS spectrometry, we can use MS mass spectrometry. Okay, then we can also use this Raman spectroscopic techniques. Okay, so this kind of detectors we may use. Now coming to the next type of gel electrophoresis, the next type of gel electrophoresis is regarded as the next type of electrophoresis technique. It is regarded as gel electrophoresis. Now in this gel electrophoresis, what happens? If you see, we are using a word gel. What is a gel? Can anyone tell me? A gel is basically a 3D matrix, okay, which are formed by forming an interconnecting solid component. Okay, in majority of the cases, you will find that the gels, they are made with polymers. Okay, it may also be made with ceramics also. So we are not considering the ceramic gels over here. So what we are considering? We are considering that we have a 3D matrix, and in this 3D matrix, it is made up of polymers. Okay. Now, these polymers, it may be a natural polymer or it may be a synthetic polymer. Okay. Incidentally, in gel electrophoresis, we have two types of gel electrophoresis. The first one is agarose gel electrophoresis, and the second method is polyacrylamide gel electrophoresis. Okay. Agarose gel electrophoresis, it employs basically agarose, which is a natural polymer, whereas phage gel electrophoresis, it uses the polymer polyacrylamide, and it uses basically uses a synthetic polymer. Synthetic polymer we use in phage. Phage stands for polyacrylamide gel electrophoresis. Okay. So here we will not get this gel electrophoresis. So in the first one, we have the natural polymer. Phage in itself is polyacrylamide gel electrophoresis. So hence I have cut this. So please note it down. So agarose gel electrophoresis and phage. Phage means polyacrylamide gel electrophoresis. In both the cases, this agarose gel electrophoresis, it is basically used for nucleic acids including nucleic acid analysis, including DNA and RNA fragments. Okay, whereas phage polyacrylamide gel electrophoresis, it is used for proteins. Okay, and this phage, it gives a very good resolution of the proteins. Okay, now the question comes, it gives a very good resolution, so why we cannot use DNA and RNA in this thing, the analysis of the DNAs and the RNAs using the phage? Okay, now if you see, the DNA and RNA, they are having very longer lengths, and the base pairs, it is in millions. Okay, so in that case, what happens is that you cannot make these polyacrylamide gels below a critical limit. Okay, I mean to say, suppose we use a polyacrylamide gel using a 4% polyacrylamide. Okay, the interaction goes in and it forms a polyacrylamide gel. However, for DNA and RNA, we need bigger pores. Okay, now if you need a bigger pore, you need lower amount of polymer within the gel. So in that case, suppose you decrease the polymer concentration to 1%, it is possible that 1% polyacrylamide gel, it will not form the gel. However, that concentration, theoretically, that concentration is needed to make a polyacrylamide gel which will form sufficiently bigger pores so that we can segregate or separate the DNAs and the RNA fragments. Okay. So due to this reasons, we need we use proteins for we use this page for proteins, and we use agarose gel electrophoresis for DNAs and the RNAs. Now how it happens? When you talk about this gel electrophoresis, as I told you, we need a polymer matrix. So in gel electrophoresis, what happens? In agarose gel electrophoresis, what happens? We have agarose, okay, which is a naturally available this gel system or the biopolymer. Now it is being dissolved in a buffer in which you want to perform the electrophoresis, okay, and it depends upon what is the pi of your protein, okay, what is the pi of your protein or the the DNAs. Inherently the DNAs, they are negatively charged, okay, the DNAs, they are negatively charged. Now since they are negatively charged, automatically you may prepare the agarose gel in neutral pH. Neutral pH means 6.8 to 7.2 pH. We may prepare the agarose gel. Now how to prepare this agarose polymer? It is taken in double distilled water or the deionized water, okay. Now the deionized water, it is having a temperature of say 40 degree centigrade, okay. At that temperature, this agarose, it gets dissolved in water. Then there are gel casting systems where you cast the gel. So what are those? Those are basically two glass systems you are having two glass plates like this. So there is a distance between that. So you stack both of them one over the other, and then what you do is that you seal off these areas from three portions: right side, left side, and the bottom. So you seal off the stuffs. Now while you are sealing off, you include a spacer. Spacer means, suppose I am having these two elements. Now if I to make it what you call in specific distance apart, what I will do, I can put my finger in between and I can put the other slide. Now in this condition, my finger, it would be regarded as the spacer. So once you have the spacer, and if we add a this plane like structure, suppose like this, if I use any material which will be helping us in keeping the what you call these two plates at equidistance from each other throughout throughout our polymerization process, then I would call that material the third material which I have used, that is a spacer, okay. Now after putting the spacer at these three sides, left side, right side, and the bottom, we seal the three sides. Okay. So after we do this, what happens? We have a 3D containerized structure where we can pour a liquid polymer solution. Now then what we do is that we pour the liquid agarose solution, and then we, after it is being put up over here, so it we fill up. Okay, another important thing, I forgot another. Yeah, we have to put another comb over here. So what is a comb? Comb is nothing but, it is similar to spacer, but it will be having some teeth. Okay, so we will put this comb over here. This comb is slightly shorter than this glass plate length. Okay, so what happens when we put this thing? So we have a place from where we can put the agarose solution. Okay, now when we put this agarose gel over here and then we keep it for solidification, when you keep it at room temperature, it will slowly solidify and it will form gel. Then after that, if we remove this comb, what will happen? We'll be getting what? We'll be getting some wells, and in this wells we need to put our samples. Okay, we need to put our sample. Now the way what I have shown you, this is in the vertical direction. Okay, this is in the vertical direction, like in this way. If you want to keep your run your gel in a vertical direction, then you put the spacer in this way. Otherwise, if you want to make the gel and you want to run it in a horizontal way, so you need to make it in this fashion. So in that case, what you will have, you will be having a container like this, and over here you will be putting the stuff. So you are having the teeth. So if you pour the agar solution over here, it will be filling up this whole container. Okay, so in between, so after you move out, so from the top you will be finding that the wells are being formed like this. Okay, and the full matrix, it is made up of the agarose gel. Okay, so once we prepare this agarose gel, then we apply the electrical field, which helps us to separate the thing. So yeah, what happens? Another important thing is that once you make the gel, you have the wells. In the wells, you need to load the sample. Okay, so whatever your solution is there, DNA mixture or whatever is there, you take that, you then put say 100 microliter of your DNA samples into each of the wells, and those 100 liter of the DNA samples, they will be having some colorants itself within the loading solution. Okay, then after you have loaded the wells with the fragments of the DNAs and the RNAs, and then you apply the electrical field, and in that way it will happen. Now can you tell me, suppose you are having this gel over here and you have the wells like this, so which one will be the anode and which one will be the cathode? In agarose gel electrophoresis, what we have told, I have told you that it is used mainly for the DNAs and the RNAs, and DNAs and RNAs, they are inherently anionic in nature. These are inherently anionic in nature. Now, in which direction we want the separation to happen? We want that the separation should be away from the well, right? So what we should have? So anion, it should move towards the anode, and here we will be having the cathode. Okay, anion moving towards the anode. So the bottom would be the positively charged terminal, and this negatively charged terminal, it will give you an electro-repulsive force towards this anion, and that will help us in the separation of the this analytes within the gel. Now you need to remember is that when we talk about agarose gel electrophoresis, it happens in the horizontal way. Agarose gel electrophoresis, it is being carried out in the horizontal platform, whereas page, they are carried out in the vertical platform. Okay. DNA gel electrophoresis, that means the agarose gel electrophoresis, it is usually performed for analytical purposes, often after amplifications of DNA by polymerase chain reaction. So you want to see, or you want to analyze whether a certain fragment of the DNA or the RNA it has been produced. Okay. Now types of gel, I already told you the polyacrylamide gels are usually used for proteins. However, they have a very high resolving power for DNA fragments in the range of 5 to 500 base pairs. Agarose gels, on the other hand, have have lower resolving power of DNA, but have a greater range of separation, and this is what we need. While you are purifying a system, you need a greater range of separation. So what does this mean? This means, suppose you are having an agarose gel and you are analyzing this say the DNA fragments. In case of agarose gel, the bands will be formed at a larger distance. However, in the case of polyacrylamide gels, the resolving power, it would be high, but they will be very close to each other, and if they are very close to each other, the extraction of those system would be very different, and many times they overlap with each other. So the separation is not that great. So what we need when we want to extract something, we need a greater range of separation. So the DNA molecules, they should be quite a distant apart. Okay, so this is why I told you that page, it is having a very good resolution, but if you want to separate these DNAs using the polyacrylamide gel, then what we will be having, it will not be able to separate the system, and due to this reason, the polyacrylamide gels, they are usually not used for the DNA and RNA separation, and they are mainly confined to the proteins. Okay. However, the resolution of over 6 MB is possible with pulsed field gel electrophoresis, which is regarded as the PFGE. Till now, we have discussed about applying a field, electrical field across the gel. However, that field it is a fixed field. That means we apply a voltage of approximately 120 volts or 240 volts DC. Okay, so these are DC electrical fields that we apply. Now if you want to use this a better resolution, then in that case we can go for the pulsed field gel electrophoresis. So in the previous case we will be having over the time, if you plot the electrical field, it will be constant. However, in the pulsed field gel electrophoresis, at an initial stage we will be giving a pulse of say 120 volts, then for a particular time it will be off, then again for some time we will be giving another pulse and there will be a off time, and after the off time again we will be giving some electrical field, and this will continue. Okay. Polyacrylamide gels, they are run in a vertical configuration, as I told you. Agarose gels, they are typically run in a horizontal fashion in submarine mode. What do you mean by submarine? Submarine means it will be within the buffer liquid. In this case, the electrophoresis liquid phase, whatever we are discussing, it will be within that liquid phase. Okay, so under the buffer, it should be under the buffer. Okay. So polyacrylamide gel electrophoresis, it is used for separating proteins ranging in the size range from 5 to 2000 kD. Okay. And as I told you, agarose gel, it sets thermally. It is a naturally occurring polymer and you do not need to perform any chemical reactions. While for developing the polyacrylamide gels, we need to perform a chemical polymerization reaction. Okay, chemical polymerization reaction. Now, gel conditions, there may be two conditions: the denaturing gel. Okay, the denaturing gel, the example of the denaturing gels is the SDS phase. Is the SDS phase. Now why? What does it does? Okay, the thing is that when you talk about proteins, I told you that phase, it is basically used for proteins. So SDS phase, it will basically be used for proteins itself, right? Now why we have added SDS? Okay, what is SDS? SDS is sodium dodecyl sulfate. It is a surfactant. Okay, it is a surfactant. Now why we use this sodium dodecyl sulfate? The reason being, over here we have proteins which is having its secondary or the tertiary structure. So they are formed in a particular orientation. So this protein, it may either be negatively charged depending upon which amino groups it is presenting on the outside, or it may be a positively charged protein, but we don't know. Okay. However, we are mainly interested in the molecular weight of the proteins. Okay. Now when I talk about the molecular weight of the proteins, so what we have to do, we have to break down this secondary or the tertiary structure, and we need to convert this into a primary structure. Now how we can do it? So we can do it by breaking the, क्या हुआ, बहुत हंसी आ रही है? So what we can do it over here is that we can apply SDS, which is a surface active agent. So what it will do, it will try to break down the hydrogen bonds, and what it will do additionally, it will help us in breaking the hydrophobic bonds. Hydrogen bonds, they are available for the hydrophilic functional groups, and hydrophobic bonds, they are present for the hydrophobic domains. Okay. Now, when you are using the sodium dodecyl sulfate, what it is doing? It is interacting with both the hydrophilic and the hydrophobic groups, and it is trying to break the this secondary or the tertiary structure. Okay. Apart from this, we have disulfide bonds, SS bonds within the proteins. Now, to break these bonds, we use reducing agents. We use reducing agents such as beta mercaptoethanol or dithiotretol. Okay, so these two are the reducing agents, and these two reducing agents, they can help us in breaking up this disulfide bonds and converting it into the SH bonds. Okay, so in this way we can break down this protein structure and bring it into the primary structure, and the primary structure, within this primary structure, this, suppose we are having this protein structure, this protein primary structure. Now this is further being stabilized by the SDS. So what it does is that it provides a negative charge over the proteins. Okay. Now, while you are doing this, in the case of agarose gel electrophoresis, what I have told you, in the case of agarose gel electrophoresis, I have told you that the DNA molecules or the RNA molecules, they are inherently electronegative in nature. That was not the case for the proteins. It may have been positively charged, may have been negatively charged. Okay. Now, by performing this denaturation, what we have done, you have converted a protein molecule into its primary protein structure. Now, if you perform the gel electrophoresis, what will happen? Their separation, it would be based on what? Based on the length, or you can say the size. Okay, because now you will be converting all the proteins which will be having the negative charge. Okay, so all of them, they will be having the same charge. So even though you are using the electrical field for the separation of these proteins, what you are basically doing it, you are basically separating the proteins based on their sizes. Okay. So the same kind of thing, it can also be performed for the nucleic acids, which are usually being denatured using the urea. And when you do this, the denaturing conditions are necessary for proper estimation of the molecular weight of the RNA, because longer your size, the higher will be the molecular weight. Okay. So urea, DMSO and glyoxal, they are the most commonly used denaturing agents for the rupture of the RNA structure. In the native gel electrophoresis, native means original form. Okay. Native means original forms. In the original forms, what you have, you do not have to add any kind of denaturing agents. So the separation, it would be based on charge, shape, size. Okay. Suppose there are two proteins or two nucleic acids which are having similar molecular weight, but they may have separate size. One may be spherical in nature, the overall structure, it may be spherical in nature, the other may be somewhat oblong in nature. So depending upon the charge, what is the charge, whether it is a positively charged analyte, whether it is its shape is spherical, whether it is an elongated form, based upon the shape also it will happen, and apart from that the size also, it would also matter. The bigger the size, the lower will be the mobility of that analyte within the gels, even under the electrical field. Okay, so this is basically the difference between the your native gel electrophoresis and the uh, denatured gel electrophoresis. Okay, here also if you see, for the biological samples we may have to use detergents. However, the use of these detergents, they are only used to the extent that they are necessary to lyse the lipid cell membranes in the cell. Okay, so if you have to extract the nucleic acids like the DNAs or the RNAs from the microbes, what you want to do, you just want to disrupt the outer cell. You do not want the denaturing agents to interact with your nucleic cells or nucleic acids like the DNAs and the RNAs and or the proteins. Okay, so you do not want that to happen. And in protein electrophoresis, what you will be doing, if there are certain intracellular proteins, then by using these detergents you can break up the cell membrane so that the intracellular proteins, they can come out, and once they come out you can go for the separation. Okay, now percentage of agarose gel and the DNA size resolution, it is given. So if you see, as you are going from top to bottom, we are increasing the agarose gel concentration for making the agarose gels. Okay, whereas in the DNA size resolution, if you see, as we are going towards the higher this agarose gel concentration, we are basically having a reduced size of the DNA. Why? I told you before also, as you increase the concentration of the polymer, your pore size, it will decrease. Okay, so what we are, these are basically these polymeric architectures they are doing, as I told you before in itself, that they are forming a 3D mesh like structure. So you are having a 3D mesh, right, like this one. Now when you are increasing this concentration, your volume is same, right? Now within this volume, what would happen? This mesh size, they will, this pores, if you see, previously it was quite bigger, now it has become smaller. Now if it has become smaller, then it will be blocking the larger particles. Okay, so the larger analytes, they cannot be used for the separation process, and hence we have depending upon uh, uh, what is the size of the DNA fragments you are expecting in your analyte, you have to choose the specific agarose gels. Okay, now next, coming to the gels, suppose you are having two gels, 1% gel and the 2% gel. I told you we can make different wells. In this case, if you see, there are only two wells. The first one is the lane 1 and the lane 2. In the lane 1, which is the leftmost well, it is used for developing ladder. What is a ladder? Ladder, whenever you do any kind of experiments, you need control. Okay, so the ladder will be made using a commercially available mixture of DNA fragments, okay, where you will be having DNA fragments of different base pairs. Okay, say for example, here we have 100, 200 and 300 base pairs. Okay, in lane 2 you will be having your own mixture. Okay, now if you see, when we used 1% gel, so what has happened? We were able to create a ladder which were having sufficient separation. Okay, however, if you look into the gel, your sample, so what was you are doing? You can see that there is only one fraction. There is only one fraction. However, if you see, when we increase the concentration, gel concentration over here, so what has happened? The separation over here, it has decreased. The separation between the 100 base pair, 200 base pair and the 300 base pair, it has decreased significantly. Okay. However, on the contrary, if you see that the separation within your analyte has happened, that means your analyte, which we were initially telling when we were running this 1% gel, we were telling that there was only one fraction. It is basically composed of two fractions. Say if we call it as A, then this A is made up of B plus C. Say this is B and this is C. Now if you see over here, so what should be the your molecular weight? Now if you see over here, we told it that this is for 100, 200 and 300 base pairs. Now this is getting out of the range. However, we can make an estimation. Suppose it may be you may be having 50, then again you may be having 25, then you can have say 10. Say for example, I am giving you a hypothetical case. So we can interpolate from here and we can say that our sample, it may be having this molecular weight or the base pair size in between say 10 and 25, say for example. However, that is not the actual way. We can estimate from here. After getting an initial information, we have to choose a ladder which encompasses our sample DNA fragments. Then only we will be able to measure the thing. In UV spectrophotometry, if you remember in your laboratory class, what we how did we make the standard? We were having the concentration, known concentration within this specific region, and we are having the absorbance. Okay. Now say this was capital X, say x0, x1, x2, x3. We are having like this, right? Now if you remember, we gave you an unknown concentration which lies within x0 and x4, right? Suppose if it was outside this range, then we needed to make another standard curve which would have taken another at least 2 or 3 concentration outside this x4 range, and that would have encompassed the concentration of the analyte which we wanted to analyze. Say for example, let us take it as let us take some arbitrary values over here. Say this was 0, 10, 20, 30, 40. We are having like this. Okay. Suppose in our case it was 45. So for analyzing that, we needed to have another concentration value in the standard curve such that it would accommodate the absorbance values corresponding to the 45 value. So say for example 50. Okay, then only you have been able to analyze the concentration of the 45 concentration and its corresponding absorbance. Okay, or further what you could have done, you could have diluted this solution and bring the concentration within this range. Okay, the same thing happens over here also. When you are choosing the ladder, you need to be careful that the ladders or the original forms what or the original fragments or the standard fragments what you are getting, they should be encompassing your standard samples. If they are exceeding, then you have to change the ladder. Okay, so herein if you see, as I told you, we could resolve the pin. So what is happening over here, as I told you, as you go towards the higher gel concentration, your resolving power, it reduces for the lower fragment or lower base pair molecules. Okay, lower molecular weight molecules. However, for the larger molecular weight systems or the analytes, there would be an increase in the resolving power. Okay, so you need to do several trials to find out which one would be a better one for your analytes. Now coming to the this overview of the gel electrophoresis, how to do it. So usually I told you that all these systems, they are being performed in a buffer. So what should be the buffer? Buffer should be the having a pH of 8. Why 8? Can anyone tell me why 8? Why we prefer a pH of 8 while we are doing the electrophoresis? Conductivity बढ़ जाएगा. True, but why? We have talked about the PI. What is our body pH? It is approximately 7.2, 7.4 यहाँ तक रहता है. So all the proteins, whatever they will be present in our body, they would be having PIs within that region. Okay, so 0 to 14. So हमारा हमने बोला around 7.4. So it is there. Now if we use a pH of 8.0 over here, and suppose you are having a protein or amino acids or whatever, say 5.8, तो क्या होगा? In this region it is positively charged, and above this region it is negatively charged. मैंने क्या बोला था? Whether it is top to bottom or left to right, near the sample, towards the bottom, anode. So when you use a pH of 8, you are basically ensuring that your proteins, nucleic acids, whatever you are analyzing, they are sufficiently negatively charged and you are able to resolve the gel properly. Okay. So the preferred buffer is TIE buffer. TIE means tris acetate buffer. It also contains EDTA. And the second buffer is TBE buffer, tris borate EDTA buffer. Okay. However, we use tris acetate buffer commonly as compared to the TBE buffer. Why? The reason being, the borate, it reacts with the sugar which are present within the this nucleic acids, and they can interfere with the analysis. Secondly, it also reacts with the glycerol which is present in the loading suspension. I talked about the loading suspension. Loading suspension, what is the loading suspension? Loading suspension is the liquid where you are having the protein or the nucleic acid being suspended within a solution, and you take up the liquid and you put it in the well. So that is called the loading suspension. So glycerol is present within the loading suspension, and finally it also reduces the activity of the loading dye. What is a loading dye? There is a dye which is also added into the loading suspension so that you can have uh, this loading suspension. You can tag these analytes with some dye so that when you are running the gel, if it is invisible, what would happen? You will not know at what position your band has reached. When you add this loading dye, when these nucleic acid fragments or the proteins, they are moving, they have become colored basically. Now you will be seeing that there are color bands which are moving, and you will know at what position it should be the right time for you to stop. When to stop? Suppose in the ladder this is the lowest in the ladder this is the lowest base pair. Okay, either when it reaches towards the bottom means approximately 1 inch from the bottom, or your analyte it reaches towards the bottom, that is 1 inch gap from the end of your gel. So when either your standard or your analyte it has reached towards the bottom of the gel, then you will find that it is the right time to stop the electrophoresis. Then at that time you will be stopping the electrical field. Okay, so we'll be continuing this discussion tomorrow.